All right, let's jump right in. Welcome to this explainer, which we've tailored specifically as a briefing for fellow molecular biology researchers. Today, we're unpacking a really fantastic 2025 study from Molecular Cell by Haija and colleagues. We're going to see exactly how nuclear compartments act as the master controllers of RNA maturation. Mapping RNA kinetics in specific membraneless compartments has always been a huge methodological headache for us, right? Well, today, we're going to walk chronologically through a novel solution. First, the SLAM-RT&Tag protocol. Then, we'll dive into transcript kinetics, look at Polycomb domains acting as gene sequesters, explore nuclear speckles as splicing checkpoints, and wrap up with how spatial compartmentalization dictates RNA maturation. So starting with Section 1, the SLRT&Tag protocol, adapting SLAM-seq for compartment-specific RNA profiling. To really appreciate the data, we first need to understand the tool. SLAM-RT&Tag is a kinetic labeling strategy that ingeniously combines RNA metabolic labeling with the spatial precision of RT&Tag. If you want to implement a step-by-step pipeline for this in your own lab, the study outlines 5 critical protocol modifications. First up, you initiate an S4U metabolic pulse to label newly synthesized RNA in living cells. Second, and this is key, nuclei isolation must happen under highly precise cold temperature controls to lock in those delicate spatial relationships before you tether them to magnetic concavalent A beads. Third, the alkylation step demands incredibly strict chemical control. You've got to use iodoacetamide or IAA at optimized concentrations to wipe out any off-target background noise. Fourth, your permeabilization buffers have to be strictly adjusted to maintain nuclear integrity. And finally, reaction timing is tightly constricted to grab perfectly accurate kinetic snapshots. It's this highly controlled pipeline that lets us trace local transcript dynamics with just unprecedented spatial precision. And this is where it gets really clever. By using highly specific antibodies, the protocol captures transcript behavior that's localized exactly around a distinct nuclear epitope of interest. You're basically creating a targeted radius, which lets you profile the transcriptome composition of one very specific neighborhood within the nucleus. Moving right along to Section 2, transcript kinetics and half-lives, revealing transient interactions. Now, what SLAM-RT&Tag uncovered completely redefines our understanding of how long transcripts actually hang around chromatin. Using a kinetic modeling tool called BACR, the team found that the median localized half-life of transcripts adjacent to Polycomb marks is 1.6 hours. To put that into perspective, their overall global half-life is 3.2 hours. These localized half-lives on chromatin are noticeably longer than what previous global estimates told us, and that's likely just because of the sheer time it takes to transcribe these exceptionally long genes. But the data presents a really sharp dichotomy. For the vast majority of these localized transcripts, we see very transient kinetic behavior, meaning they get processed or degraded locally and then they're gone. But this stands in stark contrast to exceptionally rare, persistent compartment markers. A prime example here is the mature XIS transcript, which is highly stable and remains securely locked onto the Polycomb-marked X chromosome. Which brings us to Section 3, Polycomb domains, sequestering genes, a safe harbor for transcription. Let's break down what's actually happening at these chromatin domains. Think of polychrome domains as sequestering safe harbors. The study points to a fascinating evolutionary positioning at play. Genes with exceptionally long introns take a massive amount of time to synthesize. By positioning themselves in your polychrome domains, which are typically located far away from dense processing centers, these slow-to-transcribe transcripts are essentially shielded. They're kept away from high concentrations of splicing factors, which completely prevents premature, non-canonical, recursive missplicing. And the SLAM-RT&Tag data makes it clear. Rather than housing internal transcription directly inside the dense core of the domain, these domains actually serve as protective adjacent zones. The long gene bodies and structural loops are slowly synthesized while safely anchored just outside the Polycomb mark, totally isolated from that aggressive processing machinery. To definitively prove that this physical proximity is vital for splicing efficiency, the researchers perturbed the system by knocking out SUZ12, a core component of the Polycomb repressive complex 2, in K56 cells. The fallout was dramatic. It triggered over 1,000 differential splicing events. Specifically, they observed a massive 10% rate of exon skipping in Polycomb mark-adjacent transcripts, compared to just 3% globally. So if you lose the structural integrity of this domain, you severely compromise proper RNA maturation. But this raises a really critical question for our field. Once a long transcript finally finishes synthesizing and actually leaves the protective harbor of the Polycomb domain, where exactly does it go if its introns aren't fully processed co-transcriptionally? That leads us right into Section 4, nuclear speckles, splicing checkpoints, the post-transcriptional quality control. Unlike Polycomb domains that act as a shield, nuclear speckles play by a completely different set of rules. Speckles are dense, packing a very high abundance of localized splicing factors, and they function as rigorous post-transcriptional quality control checkpoints. Transcripts that migrate here typically have fewer but significantly longer introns, which we call detained introns, or DIs. Because these DIs are structurally complex, they demand incredibly slow splicing times. We're talking about pausing in the speckle for an hour or more before they're fully mature. So there's a very clear structural association happening here. Transcripts that fail to splice co-transcriptionally migrate specifically to these dense, splicing factor-rich speckles. That high factor density is exactly the environment needed to slowly and methodically process those stubborn, delayed introns. And finally, Section 5, compartmentalization and RNA maturation, the ultimate splicing control strategy. When we contrast these two distinct environments, the overarching strategy of the nucleus becomes crystal clear. Polycomb domains act co-transcriptionally. They offer low splicing factor density to keep genes with exceptionally long introns safe for about 1.6 hours. On the flip side, nuclear speckles act post-transcriptionally, providing a high factor density for transcripts lugging around detained introns that require about an hour of delayed processing. What this directly shows us is that proper RNA processing is absolutely not just about the genetic sequence itself. It is heavily, heavily dictated by spatial biology. The specific compartment a transcript is exposed to ultimately decides its retention time and its final splicing fate. I want to leave you with this defining quote directly from the study. Nuclear compartments contribute to RNA processing by exposing transcripts to specific splicing factor concentrations. So, as you head back to the bench and start thinking about applying SLAM-RT&Tag in your own research, ask yourselves, what hidden kinetic life cycles might the transcripts in your favorite nuclear compartments be experiencing right now?